The last topic in the chapter is talking about membranes and osmosis. So the structure of the cell membrane was first proposed back in 1972 by Singer and Nicholson. And what they proposed is what we still feel today is correct, is what we call the fluid mosaic model of the cell membrane. Cell membranes are what we say as selectively permeable. They're selectively permeable because of their structure. Basically, the cell membrane in the Singer-Nicholson model was proposed as a lipid bilayer. The lipid bilayer is a two-dimensional fluid that acts as a solvent for some chemicals. It turns out phospholipids can move rapidly, so this is a phospholipid bilayer membrane. The phospholipids can move horizontally in the membrane very rapidly, and they can also flip from one side to the other side pretty quickly. Each phospholipid consists of a phosphate head. This is the phosphate head. There's a nitrogen group on here, and these fatty acid tails. So this is a phospholipid up here. So this whole region up here is actually the nitrogen compound and the phosphate. And then these fatty acid things sticking down here, these are nonpolar fatty acid tails. So let me erase this. These little blue heads here contain choline and a phosphate group, which are polar, and they want to face water. Out here is water. In here is water, which is polar, remember. The fatty acid tails that are sticking down here, those are nonpolar. So phospholipids in cell membranes are bilayers. Two layers of lipids with the phosphates facing outward and inward and the tails facing together. This is called the core of the membrane. The core of the membrane is the only significant nonpolar region in a cell. And cell membrane material surrounds most of the organelles in a eukaryotic cell. So the core of all the membrane areas in the cell is where nonpolar materials like steroids and cholesterol and other nonpolar materials reside. So the cell membrane is divided on two areas based on polarity: the phosphates, which are polar, and the core, which is nonpolar. So I've talked all about that. Time to move to the next section here. Now, lipid bilayers are fluid. What that means is they can move and stretch under stress. Cholesterol, which in this diagram, these are cholesterols here. Cholesterol, cholesterol, cholesterol. Cholesterol strengthens the interaction of the hydrophobic hydrocarbon ends. The slower moving proteins of the membrane are of two types. Some have both hydrophilic and hydrophobic areas. This is a protein with a hydrophilic, polar, polar, middle section nonpolar. Okay. This protein here is there's a little nonpolar section here, the rest is polar. This protein here, middle section nonpolar, outer part polar, inner part polar. This section, this goofy coil protein, middle part nonpolar, part that's sticking out, polar. So the slower moving proteins are these blue things: protein, protein, protein. They're of two types. Transmembrane proteins, or integral proteins, are stuck in the membrane and reach through the membrane. They have polar and nonpolar regions. The peripheral proteins tend to have only small nonpolar regions and a little bit of polar regions sticking out. So the slower moving proteins of the membrane are of two types. Some have hydrophilic and hydrophobic areas and have their hydrophobic areas securely wedged in the core of the membrane. And then some of them also have exterior hydrophilic regions sticking out. Proteins do the following. Many proteins that are wedged into the cell membrane serve as transport proteins. Some of them are enzymes that actually catalyze chemical reactions. We'll learn about those in communication. Some of them are signal transducers. That's also important to communication. Some of them are important to cell recognition. Your white blood cell uses receptors. This is probably a receptor right here with that goofy carbohydrate chain hanging off of it right there. Some proteins that are on the surface of a cell membrane are for junctions. They cement one cell to another. And that's for, I'm sorry, cell attachments are those proteins. Cell junctions, or like gap junctions in heart cells, are channels which are made by proteins that go between the heart cells for rapid impulse conduction. So cell membranes are not symmetrical. Here, let's scroll this up a little bit so you can see this. Cell membranes are not symmetrical. The two sides of the membrane are not identical. This is the outside, and this is the inside. The inside has cytoskeletal proteins attached to some of these integral proteins for strength. The outside has all of these goofy carbohydrate chains attached to the proteins to act as receptors for signals or for cellular identification. Let's continue down now. What's the purpose of membranes in cells? Membranes do two things. They form a physical barrier around a cell and thus create like a zone inside and a zone outside. And they also control the passage of materials, in and out of the cell. Substances basically cross membranes in three ways. A substance can either dissolve in the membrane. It'll dissolve in the core of the membrane, and the core of the membrane of the cell membrane is shared with endoplasmic reticulum, Golgi apparatus. So once you're in the core of the cell membrane, you're quickly going to end up inside the cell. You can also be moved by a membrane protein into the cell using like a gate protein, or you can be eaten by like what is called phagocytosis. So cell membrane material is all over the inside of a cell. These the chloroplasts, these are grana in stacks, or individual pieces would be called thylakoids. That's all membrane material. This is endoplasmic reticulum in layers going around this region right here. This is probably the nucleus in the center, and this is all membrane material. Cell membrane is all over the inside of a eukaryotic cell. Its job is to compartmentalize the cell. Now, don't confuse the cell membrane with the cell wall. The cell membrane is an entirely different construction than the cell wall. The cell wall is basically just cellulose fibers, which are big rods that lay over each other and are stuck to each other by hydrogen bonding. And there's huge gaps in the cell wall to allow materials to go through. So cell walls are not selectively permeable like cell membranes. Cell walls are made of cellulose, which is a glucose polymer. Basically, the purpose of the cell wall is to protect the cell and hold it in shape and add structural support. It's very porous, so it lets everything through. Cell walls are what we build houses with. Cellulose reinforced with lignin is what is made out of. Prokaryotes, plants, and fungi have cell walls, although the cell wall of a fungus is not made of cellulose. It's made of chitin, which is the same chemical in the skeleton of an insect. But that's another story. Now, we have to talk about how materials get in and out of cells, and that brings us to diffusion. So diffusion is the movement of chemicals from where they are common to where they are rare. It's caused by random particle kinetic energy. The random motion of particles from high to low concentration is just because they're scattering out. The overall factors that affect the rate of diffusion are the gradient. When this yellow dye is first dropped in, you have a huge gradient, huge amount of dye on the top, almost none on the bottom. The molecules scatter themselves out on their own. You don't have to stir, it just takes a while. The water molecules are in motion, they bounce around, and the dye is dispersed over time. The distance they have to travel, the area that they have to cover, and the qualities of the barriers they have to pass through are all factors affecting the rate of diffusion. Most cells, to affect diffusion, have a negative interior charge and a positive exterior charge. That affects the diffusion of ions. Ions that are outside that are negative are repelled from going in the cell because the inside of the cell is already negative. Ions that are positive outside of a cell want to diffuse more into a cell because the inside is negative. They're attracted to it. That's called an electrochemical gradient or an electrical potential gradient. So ions always have two gradients, the concentration gradient and the electrical potential gradient. And both of those have to be considered to figure out whether an ion is going to go through a membrane. Let's see. Water is a tricky thing. Water molecules are really small, and there are special proteins in the membrane of cells called aquaporins to help water move through the membrane. So water molecules move through membranes by what we call osmosis. Water goes through a membrane faster than almost any other substance because it's small and there are aquaporins to let it through. Water will always go from where it's high concentrated to low concentration unless pressure can push it backwards. That's called reverse osmosis. We use reverse osmosis to make drinking water in some parts of the world. The more solute concentrated a solution is, the more hypertonic it is. The less solute concentrated a solution is, the more hypotonic it is. We use these terms to describe the tonicity of water. Tonicity describes chemical concentration in water. If two solutions are next to each other and they are isotonic to each other, then they have equal solute concentrations. What that means is that water then won't want to go either way. But that's usually not the case. Usually, there are more chemicals on one side of a membrane than another. If blue is water and red are solutes, the chemical on the left, and by the way, this is a magnified version of the chemical. There's 4 times of the chemical on the left and the right. So water is going to want to go this way. It wants to move from the side where it's less concentrated or more concentrated to where it's less concentrated. That causes the water level here to rise and the water level on this side to fall. The reason that happens is because the membrane is selectively permeable. It's letting the water molecules pass, but not the larger solute particles. The water potential of a cell is defined as the solute potential plus the pressure potential. The pressure potential is almost always 0 unless you're dealing with a syringe or something and you're actually pushing the water. Plant cells do have positive pressure potential. Animal cells generally don't. Plant cells do when they're surrounded by pure water. The cell swells and the cell wall keeps it from blowing up. Now, the solute potential is the water potential of a cell due to its dissolved solutes. That is, the tendency of the cell to gain water when separated from pure water by a selectively permeable membrane. That is what the solute potential is. Cells have to concern themselves with water because water moves through the membrane very fast. So if a cell gets placed into an environment that's extremely hypertonic, water will move out of the cell into the hypertonic environment around it. That will plasmolyze the cell, meaning it loses all of its water, it dehydrates and dies. On the other hand, if a cell is in a hypotonic situation, water will rush into the cell because the cell is hypertonic to the hypotonic water around it. If there's nothing to stop the cell from swelling, the cell will swell up, expand, and explode. We call this lysis. So plasmolysis is the shrinking of the cell. Lysis is the explosion of a cell. Plant cells have cell walls to prevent lysis, and so you can stick a piece of wood in water and the cells won't explode. But an animal that drowns in water that's no longer pumping out excess water, the cells rupture and explode as the body absorbs water. All dead bodies in water do that. That's just a natural occurrence of osmosis going into the animal cells. Now, we've already talked about osmosis. Now it's time to talk about solutes moving in and out of membranes. Turns out that in both active and there's got plants and animal cells and all cells, bacteria, everything, have ways of moving chemicals through their membranes. There are passive ways and there are active ways. Passive membrane transport systems do not require energy to move the chemicals through the membrane. These are basically proteins that form doorways through the cell membrane and allow chemicals to go in and out. It's passive because stuff can go either way. In passive transport, the membrane protein provides a means for an ion or a molecule to traverse the liquid membrane down its electrochemical gradient. So an ion always wants to be going where it's oppositely charged on the other side, and it wants to go down its concentration gradient too. It wants to spread itself out. Now, Some carriers move ions such that charge is unequally distributed across the membrane, and thus an electrical potential difference or membrane potential exists across the membrane, and I'll show you that in just a minute. Protein channels are passive, and that's where proteins form channels in the membrane and let stuff go through. This permits small solutes to get in and out of the cell on their own. Some channels are always open, they're called channel proteins. Others are closed at times, and they're called gated channels. Nerves use a lot of gated channels to deal with nerve impulses. Some of these gates will open in response to a receptor hitting a messenger. We'll talk about that when we get to the communication chapter. So, in an active transport system, which is different than a passive transport system, energy is required to pump things. Active transport consumes a lot of energy. Most of the energy right now that you're thinking with is going to active transport in your brain. Your brain cells pump minerals. Active transport differs from passive transport in two basic ways. Active transport can move substance with or against their concentration gradient. It usually moves them against their concentration gradient. Not always, though. It also requires the expenditure of energy. And the source energy for most active transport system is ATP, but some use electrochemical gradients to do their thing. Examples of active transport are pumps in E. coli, which are bacteria in your colon that move amino acids or peptides or nucleosides and potassium. Muscle cells in your body have calcium pumps. You have hydrogen ion pumps that secrete stomach acid. And those hydrogen ion pumps are so effective they can pump them so that the protons are 1 million times more concentrated in the stomach than they are in the stomach cells. There are two basic kind of active transport pump systems on Earth, primary and secondary. Primary pump systems move a solute through the membrane using ATP energy. Secondary active transport systems are called coupled transport systems. They use the energy of an electrochemical gradient to power the movement of another solute through the membrane. Our examples of both of these right down below here. So on the left-hand side, we have a primary pump. Let's erase the... Okay, good. We have a primary pump here. ATP is being consumed by this protein. The protein is actually then pulling solutes through the membrane as a result of using the ATP energy. What the ATP actually does is it just changes the shape of the protein and causes it to flip, and that moves the solutes that the protein was sticking to. It allows the solutes to get through the membrane. Over here, what's happening is the amino acids here are being brought in by a special protein as the protein is consuming the energy of these ions. So we have, I think the author has put, these are sodium ions here, and the sodium ions are being consumed by these proteins to pull amino acids through the membrane. This pump will only work if it can actually grab both a sodium and an amino acid at the same time and move them through the membrane. If the sodium runs out here, it can't do anything with the amino acids. So it's not using ATP, but the catch is that the sodiums were pumped out here using ATP. So this is why this is called a secondary pump. It doesn't primarily use ATP, it uses ATP energy secondarily. Now, that brings us to the most famous of all the pumps, the sodium-potassium pump. The sodium-potassium pump is the most studied of all active transport pumps on Earth because it's in nerve cells, animal nerve cells, human nerve cells. This pump is most famously found on neuron and muscle membranes, but it's also found in plants. Lots of cells like to pump sodium and potassium. So what the pump does is it pumps sodium out. This is the yellow thing in the middle is the pump, P for pump. The pump consumes ATP, and in so doing, it throws sodium out. When a nerve impulse comes through, the sodium leaks in. That's what a nerve impulse is, sodium leaking into the membrane. The membrane, to restore its charge, it lets the potassium leak back out. So you see what's happening here. Let's erase the R. The pump is actually pumping sodium out. The nerve impulse, sodium leaks in. Nerve restores its charge by leaking the potassium out, and then the pump puts the minerals back in their same place. The nerve impulse cannot pass unless sodium is out here. Without sodium out here, there is no nerve impulse. So the pump pumps constantly in order to maintain the membrane's ability to pass an action potential, which is what a nerve impulse is, down the neuron. What we have here is an active transport pump in the middle, and these are two gated channel proteins. This channel protein is opened by a nerve impulse. This channel protein is opened when sodium is on the inside and the inside becomes negative. That's the cue for this one, this channel protein, to open up and allow potassium to leak out. This channel protein opens in order to let sodium in and cause the nerve impulse. But we'll talk more about this in the communication chapter when we get to neurophysiology. That brings us to the last parts of the chapter here, which is exo and endocytosis. It turns out cells also have ways of moving like whole cells in and out of themselves. Endocytosis is where cells engulf like whole materials from the world around them. Phagocytosis is the engulfment of whole cells. This is most famous when amoebas. An amoeba will absorb, like here's a little bacterium. The white blood cell literally crawls around the bacteria, engulfs it into a vacuole, fuses a lysosome with the vacuole. The bacteria is destroyed. And then exocytosis occurs, dumping the broken down chemicals out. This can actually be a problem because sometimes the exocytosis includes a toxin which is dumped out, and then it poisons the rest of your body and kills you. That happens in some bacterial infections like meningitis and things. So there's also receptor-mediated endocytosis. That's similar to phagocytosis, but instead of being whole cells, it's just clusters of molecules that are being engulfed. In exocytosis, which is the opposite of endocytosis, materials are ejected. This is showing endocytosis, specifically phagocytosis on this side, and this on this side is exocytosis. Materials are being ejected. On these two animations down below, we have exo and endocytosis being animated. On the left, we have exocytosis. We have the ER manufacturing proteins. Actually, it's the ribosomes that make the proteins. The proteins go through the ER, come off on little vesicles, they go to the Golgi. The Golgi that, okay, I shouldn't have clicked there. Click. The Golgi then, let's get my pen. The Golgi then passes or modifies the proteins and then passes them to the cell membrane, and then they're exocytosed out here. This is exocytosis that's happening here. Over here, this is phagocytosis. The bacteria, well, actually this isn't a bacteria, this is a little ciliate. Swimming along, runs into a white blood cell. The white blood cell surrounds it, engulfs it, fuses a lysosome with it, destroys it, and then the vacuole is reverse phagocytized, which is exocytosis. It belches out the waste material, which again sometimes can be a problem because in a serious bacterial infection, the poisons that are dumped out can actually kill you, especially destroy your kidneys when your kidneys try to filter them out of your bloodstream. The last topic in the chapter is trying to hammer home the idea that eukaryotic cells are really, really into membranes. Compartmentalization is a big deal. Eukaryotic cells use membranes to maintain specialized regions. Internal membranes facilitate all kinds of activities. For example, a mitochondrion is a membrane enclosed space where you burn your food. Vacuoles store food. That's a separate membrane place. ER membranes tend to hold proteins and then put them into little bubbles that go to the Golgi apparatus. The Golgi apparatus is membranes where proteins get modified so that they can go to the cell membrane and get ejected from the cell. So membranes isolate enzymes. They provide enzyme attachment points. They can hold in DNA and protect it. They play a role in protein synthesis because ribosomes on the ER membranes push their proteins through the ER membrane into the inside of the ER, so the ER can then move the proteins towards the Golgi through what are called little vesicles. So membranes are kind of a big deal. They transport things around the cell. They're involved in cell communication, etc., etc. Some organelles like chloroplasts and mitochondria are membranes inside of membranes. And then furthermore, modification of proteins requires the interaction of membranes, ER membranes transporting proteins to Golgi membranes, which will modify the proteins and send them out of the cell. These little carbohydrate chains that are stuck on the proteins there, those were put there by a Golgi. That's the Golgi's job, is to build complex proteins. Ribosomes make basic proteins. Golgi turn them into complex proteins. So glycoproteins and lipoproteins are proteins that have either carbohydrates or lipids stuck to them. They're both manufactured by the Golgi apparatus. And that brings us to the last section here. I already went over this before in the animation, but ultimately the ER membranes package proteins that are stuck into them by the ribosomes that are studded on them. Those proteins go through vesicles to the Golgi. The Golgi modifies them, adds carbohydrate chains, adds lipid molecules. That's how LDL is made. Low-density lipoprotein is a Golgi product. LDL and HDL, which are your cholesterol carriers in your blood, source or come from Golgi apparati in cells of your body, like your liver. Polypeptides are made by ribosomes. Ribosomes produce proteins. Ultimately, if the ribosome is on the ER, it pushes the protein into the ER. The ER then sends it to the Golgi. The Golgi will modify that protein, and the protein will then end up going out of the cell. Now, I believe that concludes. Yes, it does. So that concludes the matter and energy unit, which is one of our longest units of the school year.